Welcome back. So, we are discussing about those electron transfers following photo excitation. So, if we had P 700 starred, that means we have the excited species at the photosystem 1. So, this particular one is residing at PS 1. So, at PS 1 we get P 700 starred. So, that P 700 starred we have some electron from there, and at the next point it is reacting with the ferredoxin. What we have shown in the Z scheme is P 430. It is very interesting to label all these species. P stands for your pigment and this is the corresponding wavelength, because whatever species you are handling, that means where you have this Fe4 S4 species as the ferredoxin, and these are very good species we know as electron transfer material. So, it can we have the corresponding two forms depending upon its E0 for the oxidation and the reduction reactions, and all these iron sulfur proteins are we know that they can span a very wide range of E0 values. That is why what we studied earlier also we know that all these ferredoxin type of molecules can be present in photosynthesis, can also be present in respiration. So, depending upon your protein environment around these cluster molecules, you can have the oxidized form where iron centers are present in the ferric form, and the reduced form where the iron centers are present in the reduced form. So, this particular one, that means this Fe4 S4, which is our well known ferredoxin molecule, and that ferredoxin molecule is coupled with that of your P 700 starred one, and this ferredoxin protein molecule is attached to this one, and this particular level which is its corresponding electronic absorption. So, electronic absorption behavior at 430 nanometer for the oxidized form, that means the ferric form of the ferredoxin molecule. Then it is coupled with some flavoprotein. Fp is our flavoprotein. These are all our well known redox mediators, because we cannot connect all these directly to the species what we are trying to attach, that means NADP plus. So, it has one particular redox potential value, it has also one redox potential value, and between these two we have the intermediate mediators, redox mediators, which can couple with P 700 star to NADP plus. And at this point when you have PS1, and how we studied these also, that means you have one such species we already just we studied in our previous 2-3 classes. We read about plastocyanin. Which are copper proteins. So, blue copper proteins responsible for electron transfer. So, this plastocyanin is taking that electron. So, electron is basically entering in PS1 from these plastocyanins. So, plastocyanins in the reduced form can put the electron to PS1, and in that particular one when you have this plastocyanin. So, sometimes we find that whether your corresponding redox couple. So, thermodynamics will tell us whether the reaction of plastocyanin, which is a copper based one, and if it is copper in reduced form, it can react with the corresponding ferredoxin in oxidized form. So, it can react with ferredoxin in oxidized form. So, this redox couple what we studied earlier also in case of cytochrome chain and cytochrome C oxidases, that whenever you have the pair of redox partners, you can find out the corresponding change in E0 values, the delta E you can find out, and the corresponding Gibbs free energy change will tell you whether your reaction is facile or not. So, in this particular case what we find that you have this plastocyanin potential is in the range of 0.37 volt and ferredoxin 1 is 0.45 volt. So, this gives us that oxidized plastocyanin, Pc is your plastocyanin in the oxidized form and ferredoxin in the reduced form, which is nothing but Fe2 plus. So, when they are basically connected, we can calculate the delta E difference of these two, which is minus 0.82 volt, and you can calculate the corresponding delta G0 prime as plus 79 kilojoule per mole. So, this particular case, that means whether you have at a particular wavelength. So, this is basically this level is your 700 nanometer wavelength, and from that wavelength and from the corresponding values for this, we find that only this much amount of free energy change is available for this electron transfer. So, some processes we can have is the favored processes and some are not favored depending upon these values, but if we just put the radiation, the illuminated version of these can change this particular electron transfer pathways in a different way, and that we can also see in case of P680 as well, where you have certain other species connected to that. In one of them is cytochrome B, then one such species which is not based on copper, but it is a quinone based species which is plastoquinone, and the corresponding redox potential is similar to that of your catechol-quinone potential, and this then clubbed with another cytochrome B type of different version. F is fuel, which is a French term for leaf, F-E-U-I-L-L-E, fuel. So, which is nothing but one French term related to leaf. So, this, and then it also connected to plastocyanin, and this then responsible for generation of chlorophyll A1 cation radical. So, all these are involved in the different types of electron transfer chain, and if we can find out the corresponding values and how this particular say plastoquinone, because we know the corresponding potential for water and oxygen couple, and if this PQ and PQH2, that means the quinone form and the catechol form PQH2 is the catechol, and if this redox couple is this much, and we know the corresponding values for O2 and H2O, which is 0.82 volt. So, in this region whether you can have, so you can differentiate out these two values and you find out the corresponding E0, and that E0 we can correlate with that of our H nu, nothing else we are doing. So, H nu for one photosystem is 680 nanometer and another is above 700. So, these are the two values only. So, at this value what we get is 176 kilojoule, and we calculate the corresponding E0 prime. From there we can calculate the corresponding delta G0 prime, and what is the corresponding value of that kilojoule per mole. You can find out that in that particular region. So, this typical excitation can give you, and you have a corresponding delta G calculated value which will be positive in that particular case. Okay. So, these all thermodynamic parameters and all these values will talk us something related to your excitation of magnesium, how we get the electron, how we derive those electrons from those centers, and how we can put those electrons to several redox mediators, say plastocyanine, plastoquinone and all these things. But what we get that the excited electron and its chemical energy we have to transfer to our CO2 molecule, and which we cannot lose any kind of energy due to molecular vibration as well as fluorescence. So, the oxidation of this water molecule, that means when we have water in photosystem II, we get something what we call also as oxygen evolving center, because from water we are getting O2 molecule. So, is known as also oxygen evolving center, where we can go for typical oxygen evolution, and is a 4 1 electron step. So, this 4 1 electron step is required, and your water we are consuming, and analysis of metal centers, because it is very easy to know that how people discovered that magnesium is present in chlorophyll and manganese is present in photosystem II. So, it is the manganese analysis. So, any manganese analysis technique, it can be a spectroscopic technique also, that identification of manganese from its corresponding atomic form will tell us that this is basically a tetranuclear manganese. And idea behind knowing that tetranuclear manganese is very simple, because we need 4 electron transfer. And in all these cases, though we all know that manganese can have the stable oxidation states from +2 to +7. So, you can argue that okay, you have one mononuclear center, you can take out one after another electron for this corresponding oxidation as well as reduction. But in all these cases, what you have, if you have a mononuclear manganese center, and if we can take out one after another electron to go for a change in oxidation state from +2 to say +6 or +3 to +7, you will encounter with different E0 values, E1, E2, E3 and E4. So, most of the cases where we find that between these two, that means the first manganese center and its second immediate oxidized form, they are interrelated, and we can write the corresponding electron transfer equilibrium in a reversible sign, and these electron transfer properties should always be reversible. That is why we always rely on something in biological system also, the corresponding measurements using cyclic voltammetry. That means, whatever you do, that means this is the anodic or the cathodic peak potential, then you get something which is your E0. That means, whatever you do, so you can settle between these two forms, one form and the immediate next one. We are not going from here to here, here to here, otherwise your reversibility will be lost. So, when we go for this oxidation for one manganese center, say if it is a manganese 3, you immediately can convert it to manganese 4, and whatever E values we get for the corresponding transfer for the manganese 3 and manganese 4. So, what happens there, that when you go and when you just oxidize it, then we reverse the course of its scanning. That means, on the electrode surface what you have say a platinum electrode. So, platinum is there and is connected. So, on this electrode surface what you have, this manganese, and manganese is formed in the plus 4. Then you switching back the corresponding cycle, the corresponding direction of the scan. So, this is the current axis, this is the voltage axis. So, voltage, say you are scanning from 0.0 volt to say 1.5 volt. Now, you are scanning back, that means 1.5 volt to you are coming back to 0.0 volt. So, now what is happening, your manganese 4 which has been produced from the oxidation of manganese 3 is adsorbed on this electrode, and that is now reduced back. So, this manganese 4 will be reduced back to manganese 3, because you are switching back. So, only some little bit difference is there between these two values, but all the manganese 4 what is forming over there will be reduced back to manganese 3. So, the stability or the lifespan or the life period for this form, that means the oxidized form, is important. So, whatever species you have, even if you just talk about the corresponding ferredoxin, one in the reduced form and another in the oxidized form, because it has a very good correlation with that of the species what we know, when well known species is the Fe4 ferredoxin molecule, the iron sulfur proteins, Fe4 S4 species. So, it has certain similarity with that manganese 4 species, and there also we get, in this particular case we get only one electron transfer. So, it is the oxidation and it is the reduction, not that you are going from a Fe4 species from 1, 2, 3 all these steps, because to stay with in the reversible domain, that whatever you are doing, that means you are just simply oxidizing one particular manganese iron center on the ferredoxin cluster. If you go for oxidizing the other centers as well, your structure will not be stable. So, you will just break the structure, you can force it, because this is electrochemical oxidation. With the help of electrodes you are forcing that oxidation, but biological reducing agents and oxidizing agents are available. It will not provide you the sufficient E0 value for this second step or for the third step. This is one reason, and another reason is that you have this adsorbed species and which is coming back, like this manganese 4 is coming back to manganese 3. But if this is not stable, that means your manganese 4 should have certain stability, otherwise it will break, that means its structure can change. It can have a chain structure if it is a cluster, a cube type of structure. Suppose if it is a 4, it is very easy to put 4 of these metal centers like this. one here, one here, one here and one here. So, you have so much connectivities. Some of these ligands, because these other coordinates are occupied by the ligands, either sulfur or in case of Mn4 we will see by oxygen. So, if they are connected for that, then the structure will change and it will not be stable enough to go back to the original position. After this settling, what you are getting back, you can compare with that of your original structure. So, if it is ferredoxin 1, after one electron oxidation, you cycle it back, you go for the reduction, you should get back the original ferredoxin reduced form, because you are doing that particular electron transfer in solution. You have water, you have some other groups also. If this has less stability, the oxidized form has less stability, it can react with some other water molecule or it can break from their structure and the iron centers, the ferrous ion or ferric ion can leach. That means, it can leave your Fe3 plus from the cluster. So, the entity that it has should have certain stability, that when you switch back to the original position, that means, when you reduce it back, the oxidized ferredoxin, when it is reduced back, it should produce the same ferredoxin reduced form what you have originally, what you have used for the oxidation process. So, if this is not sufficiently stable, what happens? How you know? This cyclic voltammogram can show you that whatever you are producing on the electrode surface, which is adsorbed there, which is not stable, which is not the same species having the same value for this, what will happen after here? That means, you are oxidizing it and it is transforming to some other species. You are getting back not with the current value of this position. So, you will be losing the current value. So, your thing will go away and basically you are getting for this, that means, this is for oxidizing this one, but this you are not getting what you are supposed to get for the reduction of this manganese 4 species. So, that is the typical signature, whether your structure is unstable, whether you are not getting back the same structure. So, if you just go for one step to this step to that means, most of the cases the safest way of thinking this particular arrangement, this multi-center arrangement, is that you can have one particular center and 4 times of that should give you some arrangement. So, this 4 manganese center, which is your water evolving center, and this then handling the water, and immediately this has attachment for one group, which is the plastoquinone, that means, the immediate species which is responsible for electron transfer, and we are utilizing photon for this. So, photo oxidoreductase. So, this nomenclature we know already, oxidoreductases. So, this is a very important thing and very simple one also, because this is nothing but your quinone. It is a big Q, it is a quinone form. Plastoquinone is nothing but quinone. So, if you have a catechol quinone conversion, so the catechol quinone type of species is responsible to interact with water molecule and which is controlled by irradiation, and we are looking for some oxidation reduction reaction. So, this is a very big name, but what are these? So, you have water. So, from the very beginning of our classes, we just talking about 2 water molecules and 2 of this quinone form, which is the oxidized form, the plastoquinone in the oxidized form, irradiate it, giving O2 plus twice of the catechol form of this. So, this particular one we get, but for that particular transfer we need to transfer 4 electrons. So, like that of your particular metal center, where your manganese is there in +3 oxidation state, and stepwise you are oxidizing by 1 electron, then another electron, then third electron and the fourth electron. So, manganese +3 can go up to manganese 7. But similarly, if we have this particular, so we need the 4 electron species, and we can have arrangement of a tetranuclear manganese complex. So, this tetranuclear manganese complex, so one particular oxidation state we can consider it as S0 state. So, S is related to the spin state. So, one oxidation state, then what people have done, that you change it to another state. So, you are just leveling, that cluster is your Mn4 cluster. So, this cluster complex, when you get this, so S0 to S1 we get through ejection of electron, that means through oxidation. So, through oxidation, so at the same time it is also liberating one proton. So, that we all know that whenever there is electron transfer, when some system is getting oxidized, it can be your amide backbone also. So, CONH backbone, when it is bound to the metal center, if you go for the oxidation of the metal center and the nitrogen is bound to the metal center, but at the same time when you take out the electron from the metal center should be electron greedy, and that will go for the deprotonation of the amide function. So, your CONH will be CON minus. So, that can be very readily stabilize the high oxidation state. So, whatever you have in the system, when you go for 1 electron oxidation, simultaneously you will be losing 1 proton. It can be from any other bound ligand, or it can be from sometime from the bound water molecule or the hydroxide group also. So, this particular thing is activated by, because all the steps are activated by H nu. So, these are the steps like the dark phases and light phases of photosystem II. Knowing this particular photosystem II for water oxidation, whether it has to be illuminated or not, that we should know. So, at this particular step you put H nu and you get S1, and in the second step we have other step, that means S2, and S2 is again by loss of second electron, and it is also triggered by H nu, but no proton loss at this step. So, this can be differentiated from your S1. Then we have the S3 step, again one more electron loss and one more proton loss, and is also H nu. This S3 by simple electron loss can go to S4, so this is the step where you get this particular unit. So, you have 1, 2, 3, 4 electrons have been lost from the system. So, by doing so, that means by doing or taking the electrons one after another, we can move from S0 to S4. So, S0 you can immediately say that this is the super reduced form of this M4 complex, and this is the super oxidized form. So, either you put all together the 4 electron to the system or you take out 4 electron from the system. But interestingly, among all these 5 species, so you have 5 species and you can immediately say that this we are talking about 4 electron transfer. So, 4 electron transfer plus 1 starting species. So, 1 starting species plus 4 electron transfer giving you 5 states. So, 5 this form, and out of these form S1 is the most stable form. So, this is the stable form in the dark. And this particular one, so since it is stable, it is very easy to identify also this corresponding oxidation state of the manganese, and this particular one has been identified as a manganese 3,4 complex, where you have 2 of the manganese centers in the trivalent state and other 2 in the tetravalent state. Sorry, manganese 4 2. So, it is a mixed valent species where you have 2 of the manganese in the +3 state and 2 other in +4 oxidation state. So, basically what we are talking about here, that the most readily accessible oxidation states what we are talking about here is that it can go from 3 to 4 or it can go down from 3 to 2. So, by doing so you just this particular step you get 2 plus 2 4 electrons. So, this is the simplest possible picture what you can have, and in this step when you have this oxidized form. So, oxidized form basically will liberate 2 proton from here, and this is the steps where you get your dioxygen. So, whatever cycle we write over here in this particular step, because we are talking about only the photo excitation. You have given photon, you have taken out electron and the protons are coming out. So, this particular, so S4 basically collapsing to S0 when it is reacting with 2 molecules of water, giving you that required dioxygen molecule. So, collapsing of that thing to S0. So, now the challenge is that how we can identify slowly that what are these species. So, manganese is well known. So, manganese is there and we can have the knowledge about this manganese, that manganese has corresponding affinity for oxides, it is not a corresponding iron sulfur type of thing. So, it is very easy to know also once we know the tetranuclear ferredoxin molecule, the 4 iron ferredoxin molecule. That means, you can have these 2 plus you can have sometime loosely bound water molecules. So, these are basically from oxygen environment, and these oxygen environments are further supplemented by the corresponding protein side chains, that means the carboxy side chains or histidine side chains. So, these are the other ligating groups and those groups can give you all these values, and this particular conversion from water to hydroxido group to hydroxido to oxido group will also responsible for the elimination of your required number of protons during electron transfer. So, the very basic assembly for this tetranuclear system, what we can see now, that once you have this, that means if you just go for a mononuclear to a binuclear system, we all know that you can have some connectivity, at least one connectivity to get a binuclear one, and you can have certain connectivity such that if you have a group like this, this oxo or the hydroxide function can do very easily in a mu3 form, hydroxide of bridge mu3, that can give you a trinuclear one, and also this O2 minus, when these are there, this O2 minus, if it is bridging 4 metal centers, you get a very quickly a tetranuclear assembly. But for this sort of proton transfer, that means if you have a hydroxide of bridging or oxide of bridging, this is not a stable structure. Once you can go for protonation, immediately we lose the entire structure, because it is the group, that means the oxide of function you have, and this is basically the nucleus of the cluster, because it is a tetrahedral arrangement. This oxygen is in tetrahedral geometry, and this tetrahedral geometry is basically holding all 4 metal centers. It can be your 4 manganese also. But if you immediately go for protonation, so one protonation step for this O2 minus can change this to a hydroxide. This X is nothing but your hydroxide group, and its affinity for coordinating 4 metal center will be lost. So, it cannot be a mu4 ligand. Immediately when you go for protonation, it would be a mu3 ligand, if not a mu21. This is a mu21. So, what is the other alternative formulation what you can have to get this is, if you have this dinuclear assembly and if you put another dinuclear assembly close to it. So, that should be our right choice for a tetranuclear assembly, and that we all know comparing the corresponding Fe4S4 ferredoxine structure. So, that structure we have, and not that this Fe4 unit which can give you a corresponding manganese 4 unit, but also there are certain analysis, and that analysis tells us that this particular Mn4 is there, as well as we can have a nearby calcium ion like the superoxide dismutase or like the magnesium and all this. So, this presence of this calcium, it is not a redox active metal ion. So, it will not take part in any kind of electron transfer behavior, but this has always, you will find whenever any complicated structure you find, either it is a synthetic one or is a biological one. So, it has some important role to play in catalysis. How? Because it can have large number of water bound molecules or any other group bound molecules. So, it can provide some activated water molecules instead of free water molecule, and also it can contribute to a large extent to the structure of the system. That means, if you have a cubane type of structure or cube like structure, it has something to play with that cubane structure. So, if we level all the 4 manganese centers around this one particular central oxide of function, this is manganese 1, this is manganese 2, and the third one is manganese 3. So, this particular arrangement gives us something that it is not a typical cubane type of arrangement, cube like arrangement, but this particular one which is there as a mu3 one. So, if we have something, that means the mu3 group is there, and in a cube type of arrangement, if you have all the alternate corners are occupied by the metal centers. If it is a 4 metal unit, it can be your ferredoxin F4 or this water evolving center Mn4. So, these are the 4 metal centers, and these other groups, these are your bridging groups. It can be your hydroxide group or the oxide group, and the nature of these groups are mu3. It is all these X groups are binding 3 metal centers together. So, when they are occupying the 4 corners of the cube, they are in mu3 mode, the binding is in mu3 mode. So, we have satisfied this particular mode in the mu3 mode. So, when you have mu3 mode, and in this particular case, that if you have a system which is Mn4 and O4, forget about the nature of this O, because we get different spectroscopic analysis and X-ray structure analysis to some extent. We get this arrangement and we can differentiate only that whether these particular groups are oxygen or sulfur or any halogen group, but sometimes we are not able to precisely determine whether this is oxide group or hydroxide group, whether this oxygen is attached to a proton or not, that sometimes is very difficult to identify. So, this mu3 group, so it can be your hydroxide function or it can be your oxide function, and this mu3 group, when you are viewing from other direction, that means depending upon these, that means if you have 4 manganese center and 4 oxo center, you get the regular cube structure. That means all the 8 corners of the cube are occupied. But if you try to have something, that means 4 manganese are there, we know, but if you make it Mn4O3, what will happen? If you make it MnO3, that means your one of the vertex is missing, but still you have this cuboidal arrangement. If you take out this vertex and if your other bond distance and bond angle remains same, you will have basically a cuboidal structure. We call those structures as defective cubes or partial cube. We call them as defective cube or partial cube. So, in this arrangement, so we have these groups. So, one of these, that means if you take out this and if you view from here, if you try to view from this direction, you will find that this particular oxygen is like this, attaching to manganese 1, manganese 2 and manganese 3, viewing this one. So, this is your this one. Then you have another oxygen. So, this side oxygen, so this I made. So, how you nicely you can draw, you should be able to draw it also very nicely, that within a hexagonal arrangement. So, this is basically this one, because you have all, you have 1, 2, 3, 4 bridging atoms, 4 of these oxygens, 1, 2, 3, 4 these and 3 metal. So, you have this 4 manganese you have, and sorry, the reverse one, Mn3 O4, this one. So, whether you have taken out this, that means in this particular case you can take out one metal center or you can take out one bridging group. So, you can make it a manganese 3 system or you can make it a manganese 4 system with 3 bridging groups. So, this oxygen is there. So, if you find that this one is immediately is only mu2, and if it can extend its binding to the other direction, because it is a highly distorted geometry. So, once you start from this particular core structure, so core structure is a partial cubane structure, then you have this manganese 4, and in between these things is not directly connected, you have the calcium close to manganese 3. So, what we have in our hand is a Mn3 from this unit, a calcium, 4 oxygen and a distant manganese. So, this is your distant manganese, and this O2 little bit we should know about the environment. So, it is bound to nitrogen of histidine 332, then it is bound to glutamate, it is only oxygen and nitrogen environment, glutamate 189, and this one is bound to another carboxy function of amino acid is aspartate. (342), and since calcium is not a transition metal ion, sometimes it so happen, which is not so common for transition metal ion, that the same carboxy function like acetate function, what we all know that it is very difficult to go for a corresponding binding of acetate group to copper. Acetate group most of the time it can function as a bridging group only. So, these two oxygens, when they are utilized for coordination, it should go for two different metal centers, for copper, iron and all. But calcium, it is loosely bound because both the two bonds are not same, it is loosely bound. So, this is alanine, this is Ala 344, and this again to a carboxy end, quickly bound, and another So, this basic arrangement what we can have in our hand, that during this transformation when we go for the S4 state. So, electron transfer is taking place from one after another from this manganese, both of them are, two of them are manganese in the trivalent state and two of them are in tetravalent state. But how this calcium is playing some important role, not only for this structural change, but also for a different type of arrangement, whether this calcium can go out from another form. So, another form we can get, the another structure we will see, will not show all the detailed structure of these, how this particular arrangement can immediately change. If we can have this manganese oxygen is here, if we bring down this one over here, that basically is taking place. So, a change in the structure, and this within this hexagon we are trying to draw, but the positions are changed. That means, if you have a cube like structure and all the four positions what are occupied by metal center is now occupied by oxygen. That means, their positions are interchanged. The position of manganese and position of oxygen can change. So, that is a typical structural change for the entire complex. So, this one is connected with this central oxygen and 2 is now over here, and 3 is here. This is this oxygen, you see is not that we are making all these structures in the laboratory, but only for one simple electron transfer the thing has changed from this group to that group, where you have now close to that partial cube structure, your calcium is close to number 3 and it is within the cube structure. Now, this same calcium due to that electron transfer has come out to this position. Here is now your calcium, which was earlier close to manganese number 3. This was your manganese number 3, and this 2 is basically this group which is attached to the other manganese. So, that means this particular group Lr344. So, basically what we are now labeling that your manganese center which was here has moved from here to here. This is the manganese position now. You see, this original manganese position is now occupied by the oxygen, and oxygen position is now occupied by the manganese. So, that much change. So, basically it is moving. So, if you consider that is a clockwise movement. So, if you consider this as a regular hexagon, which all the regular hexagon we know has a C6 symmetry. So, you just simply move in a angle of 60 degree, just you move for an angle of 60 degree. But for that, the driving force for that is that now 2 is there, and people can identify not only the metal, but also these groups. The Lr344 can be identified very nicely. So, this Lr344 is not moving away much, because that is why I pointed out that this is a typical binding, typical coordination of the entire carboxy function to the same metal center, which we do not see in case of any metal salt like copper acetate, iron acetate, any metal acetates. The acetate groups are mostly bridging. So, copper acetate is basically a dimer, because your acetate groups are bridging two copper centers. So, this is rather a different type of coordination where the entire acetate group is bound only to the calcium, and this is not showing any interaction with that available group, because you do not have any manganese over here. But if you move one manganese from here to here, you have available manganese over here, and this can establish the bridging group. So, you see that is the role of the manganese and as well as the calcium. Why calcium is there, and calcium is playing something, the role of the calcium what is being played for its structural change. So, calcium was there, which is this acetate coordination. Now, calcium and this manganese is now bridged. So, whatever entity we can have, we have a heterodinuclear system, we can consider it as like the superoxide dismutase, what we know that it has copper and zinc center. In superoxide dismutases you have copper and zinc center. Now, it is a system where you have a acetate bridged calcium manganese compound. It is very difficult to make also. If you go for making some synthetic molecule out of this, if you have a binucleating ligand, and if you have a binucleating ligand like this having two different pockets, you can have a phenol unit over here, and this phenol is coordinating to calcium and manganese, and you are using some salt as the metal salt, manganese acetate as the salt, calcium acetate can also be made, but it is not possible to make this heterodimetallic system, because all the time you are will be ending with either a dicalcium compound or a dimanganese compound, because your coordination environment for these two, if they are symmetrical in nature. So, whatever binucleating system you can have, one particular part should favor manganese coordination and other particular part should favor the calcium coordination. So, that is why this sort of chemistry is pretty difficult to do, because if you have a huge database to know that what are the groups which can bind very easily to manganese center and what are the other groups which can favor the calcium binding, then only you can get a simple binuclear compound having calcium in one pocket and manganese in other pocket. But here if you have this particular group and is already available, but only thing what is happening over there, is the translocation, that means the movement of the metal center. So, within the cluster arrangement, since you are putting electron and you are taking out electron, your structure is getting changed. Why it is getting changed? It is not that you have the oxidation or reduction, you are changing what? You are changing manganese oxygen distance, you are moving from manganese 3 to manganese 4, you are going from one particular case level of protonation to other level of protonation, that also can change the manganese oxygen distance, as well as you can change the manganese oxygen manganese angle depending upon your nature of this group, whether it is oxido function or hydroxido function, as well as the nature of the two oxidation states. So, these are the other contributing factors which can contribute for the structural change, but the calcium is responsible for binding because it do not have any preference for a regular coordination geometry. That is why calcium. If you think that manganese is there or any other transition metal is there, and if they are hexa coordinated, immediately we will say no, this can only prefer a octahedral geometry, and for a octahedral geometry you know these bond distance, bond angles are all fixed. But if you go for a main group element or a non-transition element like zinc also, it has no crystal field stabilization, it has no special preference for coordination geometry. If you have a metal center like this, the calcium or cadmium, suppose have a calcium, what happens that since it has no such preference for this binding, only the available donor groups from this protein chain or some biological chain, only the available donor groups can come, and it is such that it can have all sorts of arbitrary coordination. So, this one such arbitrary coordination can make this particular group as a bridging mode. That is why you have very distorted one, because one bond is short and another bond can be long, because initially you can think of that this acetate function is coordinating to calcium center in a monodentate fashion, because these two bonds are not same. So, it is not a symmetrical binding of the acetate group as a bidentate ligand, because acetate cannot be a bidentate ligand, other it is forming a 4-membered ring. So, one bond is short and another bond is long, which is in the other direction. So, this calcium can change the structure in a large way. You have this nitrogen coordination, you have this acid coordination from the glutamate group, because these groups are not moving. This is still the glutamate 189. But what change is taking place? So, this is one part of change, that means your system is moved from this direction to 60 degree, and you have this, and this oxygen was mu 3. Now, this is not there, but what will happen? Your this manganese number 4 is there, you will have this manganese, you have this. So, earlier you have this manganese which is connected to a mu 3 oxido function. So, it will try to establish, it will try to remain there with the help of some other bridging groups. So, this is the part which is very different from the other structure. So, this particular group which was there on this number 4 can stabilize your arrangement like this. Sorry. So, you have this group, so it is moved, but your number 4 manganese cannot move that much. That is why it is a system where you have Mn3 plus 1 manganese is not, which is different from an arrangement where you have a regular cube structure. So, once it is there and you move, basically what we are moving 1 manganese over here. So, once you move that manganese, so this particular new bridging function can take place, because you have immediately you can take over water. After deprotonation you can go for hydroxide of bridging, or further deprotonation you can go for oxide of bridging. So, this particular unit, which is a special one, which can go for bridging, and this particular distance we can measure, which is 3.3 angstrom. That means, it is tightly bound to the M3 unit. It is not that very loosely bound. Earlier we can consider this is a dangling one, which is a pendant one. This fourth manganese is a dangler, that is why I call it as a dangler, but when it is form a firm bridging, it is within this cluster, and that basically tells us that how this particular one is responsible for accepting water molecule, and it goes for the removal of this O2 once you get one group of this and another group of this from the other unit to make a OO bond. So, OO bond formation can take place, and these two manganese groups basically can activate this particular oxygen and this particular O2 unit from there. So, basic idea behind this thing is that how you go for the change in structure during electron transfer. Okay.